461-0457. 11. Immunity. Immunity is the resistance against the diseases in the body. The immune system shows how cells and molecules function together to protect the body against infectious diseases and how, after an initial infection, the body is protected from subsequent infections by the same pathogen. Phagocytosis is an immediate nonspecific part of the immune system, while the actions of lymphocytes provide effective defense against specific pathogens. 11.1 The immune system. We are surrounded by potential pathogens, disease-causing organisms, such as bacteria and viruses which can cause disease if allowed to enter and multiply inside the body. The body's defense system consists of barriers to keep microorganisms out, and mechanisms to detect and destroy those that do enter. Together, these mechanisms are the immune response. The immune system is capable of nonspecific responses. Inflammation and phagocytosis occur in response to any invading microorganism. Specific mechanisms allow the body to recognize and fight individual types of microorganism. There are two specific responses: cell-mediated immunity and humoral immunity. Lymphocytes, white cells, are responsible for the specific immune response. T cells mature in the thymus gland and B cells come directly from bone marrow. Both are able to recognize foreign antigens that come from pathogens. B cells are responsible for humoral immunity. They secrete specific proteins called antibodies. T cells are responsible for cell-mediated immunity and help to control the overall specific response. Killer T cells attack pathogens or infected cells. Helper T cells activate B cells, telling them to secrete antibodies. Suppressor T cells damp down the immune response when the infection is over. Autoimmune diseases occur when the body's immune system attacks its own tissues. Examples include rheumatoid arthritis, multiple sclerosis, and myasthenia gravis. Allergies occur when the body overreacts to a normally harmless substance such as pollen. When cells or tissues are taken from one individual and given to another, they are often recognized as foreign and destroyed. This process is often called graft rejection. To minimize the chance of rejection in blood transfusions and organ transplants, antibiotics are important drugs for treating infections caused by bacteria have evolved to become resistant to them. Antibiotic-resistant strains of many bacteria are becoming common and represent a problem for doctors and researchers. 11.1 The mode of action of phagocytes, macrophages, and neutrophil. Phagocytes and lymphocytes are the cells of the immune system. Phagocytes originate from stem cells in the bone marrow and are produced in the bone marrow throughout life. There are two types of phagocytes. Neutrophils circulate in the blood and enter infected tissue. Macrophages remain inside tissues. Neutrophils and macrophages engulf bacteria and viruses via phagocytosis and destroy them by intracellular digestion using enzymes from lysosomes. Lymphocytes also originate from stem cells in bone marrow. There are two types: B lymphocytes, B cells, and T lymphocytes, T cells. B cells and T cells gain glycoprotein receptors that are specific to each cell as they mature. B cells mature in bone marrow. T cells mature in the thymus gland. As they mature, individual B cells and T cells gain specific glycoprotein receptors. During maturation, many T cells are destroyed, as they express. If left to circulate in the body, they would destroy cells and tissues. The T cells that are not destroyed recognize non-self antigens, such as those on the surfaces of pathogens. T. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Cells differentiate into either helper cells or T killer cells. Each T cell and B cell divides to form a small clone of cells that spreads throughout the body in the blood and in the lymphoid tissue, e.g. lymph nodes and spleen. During an immune response, those B and T cells with receptors that are specific to the antigen are activated. When B cells are activated, they form plasma cells which secrete antibodies. T cells do not secrete antibodies. T helper cells secrete cell signaling molecules, cytokines, that control the immune system by stimulating B cells and T cells to divide and become active. Active T killer cells can seek out and destroy any host cells that are infected by pathogens. During an immune response, memory cells are formed which retain the ability to divide rapidly and develop into active B or T cells or a second exposure to the same antigen, immunological memory. 11.1 to 2 Antigens, self-antigens and non-self-antigens. Antigens, usually proteins, trigger the immune response. Antigens are foreign, non-self, molecules that stimulate the immune system. A self-antigen is an antigen that the immune system derives from the body it is protecting. For example, a heart cell, a liver cell, and a kidney cell would all contain self-antigens. A healthy immune system tolerates self-antigens and does not attack them. Non-self-antigens include the antigen structures found on the surfaces of foreign bacteria, viruses, fungi, parasites, or other non-native biocompounds. The immune system relies on antibody-antigen binding and the binding of antigens with receptors on B or T cells to recognize non-self-antigens within the body. 11.1 to 3 Sequence of events that occurred during a primary immune response with reference to the roles of macrophages, B lymphocytes, including plasma cells, T lymphocytes limited to T helper cells and T killer cells. Macrophages are activated by membrane-bound signals delivered by activated Th1 cells as well as by the potent macrophage-activating cytokine IFN-gamma, which is secreted by activated T cells. Once activated, the macrophage can kill intracellular and ingested bacteria. B-cell development takes place in a series of 6 main stages. First, they start as common lymphoid progenitor cells, which become early pro-B cells, then late pro-B cells, next large pre-B cells, then small pre-B cells, and finally, immature B cells. Lymphocytes also originate from stem cells in bone marrow. There are 2 types, B lymphocytes, B cells, and T lymphocytes, T cells. B cells and T cells gain glycoprotein receptors that are specific to each cell as they mature. B cells mature in bone marrow. T cells mature in the thymus gland. As they mature, individual B cells and T cells gain specific glycoprotein receptors. During maturation, many T cells are destroyed, as they express. If left to circulate in the body, they would destroy cells and tissues. The T cells that are not destroyed recognize non-self-antigens, such as those on the surfaces of pathogens. T cells differentiate into either helper cells or T killer cells. Each T cell and B cell divides to form a small clone of cells that spreads throughout the body in the blood and in the lymphoid tissue, e.g. lymph nodes and spleen. T helper cells enable B cells to respond to the presence of an antigen. 11.1 to 4 Role of memory cells in the secondary immune response and in long-term immunity. During an immune response, memory cells are formed which retain the ability to divide rapidly and develop into active B or T cells or a second exposure to the same antigen, immunological memory. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. B lymphocyte memory cells are stored in the lymphatic tissue. Immunological memory is a unique property of the immune system as it can store information about a stimulus and can mount an effective response when the stimulus is encountered again. This response, a secondary immune response, is quicker and stronger than the primary response. 11.2 Antibodies and vaccination. Vaccines use inactivated virus or bacteria or part of them to stimulate the body's own immune response, including antibodies that help to recognize and destroy infected cells. Passive immunity is the introduction of antibodies either across the placenta or in breast milk, or artificially by injection. Passive immunity gives temporary immunity. Vaccination confers artificial active immunity by introducing a small quantity of an antigen by injection or by mouth. A vaccine may be a whole living organism, a dead one, a harmless version of a toxin, toxoid, or a preparation of antigens. mAbs are used in diagnosis, for example in locating blood clots in veins, and in the treatment of diseases, such as breast cancer. 11.2 to 1 The molecular structure of antibodies and their functions. Antibodies are proteins that protect you when an unwanted substance enters your body. Produced by your immune system, antibodies bind to these unwanted substances in order to eliminate them from your system. Another word for antibody is immunoglobulin. Antibodies are also known as immunoglobulins, globular glycoproteins. It is the class of antibodies that have a quaternary structure composed of two identical long polypeptides, heavy chains, and two identical short polypeptides, short chains. The two variable regions of the antibody molecule form antigen-binding sites. Each type of IgG antibody can bind to two antigen molecules. Antibodies agglutinate bacteria, prevent viruses infecting cells, coat bacteria and viruses to aid phagocytosis, act with plasma proteins to burst bacteria, and neutralize toxins. Structure of antibodies. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Structure of antigens and antibody. 11.2 to 2 Hybridoma method for the production of monoclonal antibodies. Monoclonal antibodies, mAbs, are antibodies that are all identical to each other. mAbs are produced by fusing a plasma cell with a cancer cell to produce a hybridoma, which divides repeatedly to form many genetically identical cells that all produce the same antibody. Antibodies act by agglutination, producing antitoxins, lysis, or opsonization. 11.2 to 3 The principles of using monoclonal antibodies in the diagnosis of disease and in the treatment of disease. Immunoassays use monoclonal antibodies to diagnose infections, such as human immunodeficiency virus, HIV, malaria, and chlamydia. This can make disease identification much easier. Monoclonal antibodies have been made for antigens found on the bacteria Chlamydia trachomatis, which is the cause of the disease chlamydia. 11.2 to 4 Difference between active immunity and passive immunity, natural immunity and artificial immunity. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. The major advantage to passive immunity is that protection is immediate, whereas active immunity takes time, usually several weeks, to develop. However, passive immunity lasts only for a few weeks or months. Only active immunity is long-lasting. Natural immunity is acquired from exposure to the disease organism through infection with the actual disease. Vaccine-induced immunity is acquired through the introduction of a killed or weakened form of the disease organism through vaccination. Artificial immunity is a mean by which the body is given immunity to a disease by intentional exposure to small quantities of it. 11.2 to 5 Vaccines containing antigens stimulate immune response to provide long-term immunity. Active immunity is the production of antibodies and active T cells during a primary immune response to an antigen acquired either naturally by infection or artificially by vaccination. Active immunity gives long-term immunity. 11.2 to 6 Vaccination program to help and control the spread of infectious disease. Vaccines are medicines that help to protect you and your family from infectious diseases. Vaccines help your immune system to recognize viruses and bacteria and destroy them quickly. Vaccines can give you side effects, but these usually go away quickly. When you get a vaccine, it sparks your immune response, helping your body fight off and remember the germ so it can attack it if the germ ever invades again. And since vaccines are made of very small amounts of weak or dead germs, they won't make you sick. Key words: active immunity, agglutinins, antibiotic, antibodies, antigen, antigen-presenting cell, antitoxins, artificial active immunity, artificial passive immunity, autoimmune disease, B effector cells, B lymphocytes, B memory cells, clonal expansion, clonal selection, hybridoma method, immune response, leukemia, lymphocytes, memory cells, monoclonal antibodies, myasthenia gravis, natural active immunity, natural passive immunity, non-self antigens, non-specific immune response, passive immunity, phagocytes, phagocytosis, phagolysosome, phagosome, plasma cell, primary immune response, secondary immune response, self-antigens, specific immune response, T helper cells, T killer cells, T lymphocyte, T memory cells, vaccination. Chapter Glossary. Active immunity is resistance in an organism that has developed through the production of specific antibodies in response to a pathogen. It provides long-lasting immunity as memory cells are produced. Agglutinins are chemicals that cause pathogens to aggregate together, aiding phagocytosis. Antibodies can act as agglutinins. Antibiotic is chemical or compound produced by a living organism that kills or prevents the growth of bacteria. Antibodies are immunoglobulins produced by B lymphocytes in response to a specific antigen, triggering an immune response. Antigen is a chemical present on the surface of a cell that induces an immune response. Antigen-presenting cell is a macrophage that displays foreign antigens. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. Antitoxins are chemicals produced by white blood cells that neutralize toxins released by pathogens. Artificial active immunity is the production of antibodies by the immune system following the exposure to a weakened, attenuated or dead pathogen, e.g. by vaccination. Artificial passive immunity is the immunity acquired from the administration of specific antibodies from another organism. Autoimmune disease is a condition in which the immune system fails to distinguish between self and non-self antigens, so attacks and destroys healthy body tissue, e.g. arthritis, lupus. B effector cells are the type of B lymphocytes that divide to form plasma cells. B lymphocytes are the lymphocytes that mature in the bone marrow. There are three main types: plasma cells, B effector cells, and B memory cells. B memory cells are lymphocytes that provide immunological memory. Clonal expansion is rapid mitosis of specific antibody-producing cells. Clonal selection is the identification of an antibody-producing cell with complementary receptors to the shape of a specific antigen. Hybridoma method is a method used to produce large numbers of monoclonal antibodies. An antigen is injected into a mammal to stimulate clonal expansion of complementary B cells. These B cells are harvested and fused with a myeloma, which can undergo mitosis an indefinite number of times. The hybrid cell line is called a hybridoma. Immune response is the body's response to antigens it identifies as non-self, consists of a nonspecific phase involving neutrophils and macrophages, and a specific phase involving T and B lymphocytes. Leukemia is a group of blood cancers which generally begin in the bone marrow and are characterized by an abnormally high white blood cell level. Symptoms of excess bleeding, bruising, and fatigue arise due to depleted numbers of normal blood cells. Lymphocytes are white blood cells that contribute to the specific immune response. Memory cells is a subtype of lymphocyte which is produced after primary infection and remains in low levels in the blood. These cells undergo rapid mitosis if the same pathogen is encountered again. Monoclonal antibodies are identical antibodies that have been produced by an immune cell cloned from a parent cell. Myasthenia gravis is an autoimmune disease that causes skeletal muscle weakness due to the production of antibodies which damage nicotinic acetylcholine receptors at the neuromuscular junction. Natural active immunity is the production of antibodies by the immune system following infection. Natural passive immunity is the immunity acquired by an infant mammal when antibodies are transferred through the placenta and the colostrum from the mother. Non-self antigens are foreign antigens that originate from outside the body and induce an immune response. Nonspecific immune response is the first line of defense against all non-self antigens involving phagocytosis by neutrophils and macrophages. Passive immunity is resistance in an organism acquired via the transfer of antibodies. It provides short-term immunity as no memory cells are produced. Phagocytes are specialized white blood cells that engulf and destroy pathogens. There are two types, neutrophils and macrophages. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 030046100457. Phagocytosis is the process by which phagocytes engulf and destroy pathogens. Phagolysosome is a vesicle within a phagocyte formed by the fusion of a phagosome and lysosome. Phagosome is the vacuole inside a phagocyte in which a foreign particle is engulfed. Plasma cell is a type of B lymphocyte that produces antibodies specific to a particular antigen. Primary immune response is the response of the immune system to a pathogen when it is first encountered. A small number of antibodies are produced slowly. Secondary immune response is the response of the immune system to a pathogen when it is encountered for a second, third, fourth, etc. time. Immunological memory gives a rapid production of a large number of antibodies. Self-antigens are antigens present on an organism's cells that are tolerated by their own immune system. They induce antibody formation in other organisms. Specific immune response is the second line of defense against substances with non-self. Antigens begins when complementary T helper cells bind to the antigen and secrete interleukins that trigger rapid mitosis of complementary B cells or T killer cells. T helper cells are T lymphocytes with CD4 receptors on the cell surface membrane. These bind to antigens on antigen-presenting cells and secrete interleukins. T killer cells are T lymphocytes that produce perforin, destroying pathogens with a specific antigen. T lymphocyte are lymphocytes that mature in the thymus gland. There are four main types: T helper cells, T killer cells, T memory cells, and T regulatory cells. T memory cells are T lymphocytes that provide immunological memory. Vaccination is the deliberate exposure of an individual to antigens from a pathogen to provide artificial active immunity. Short MCQs with right answers. The immune system. 1. Which blood cell type does not recognize, engulf and digest non-self particles? T killer cells. 2. Repeated infections with malaria result in more effective immunity to malaria. Which type of immunity is responsible for the more effective immunity? Natural active. 3. A successful vaccination program provides a level of immunity where the majority of a population is protected. There are several factors that can affect the success of a vaccination program. Which row correctly shows the factors that can affect the success of a vaccination program? 4. Monoclonal antibodies are produced using the hybridoma method. What are removed from the spleen of a mouse to produce monoclonal antibodies? Lymphocytes. 5. Which processes characterize the mode of action of phagocytes? 1. Receptor binding. 2. Endocytosis. 3. Exocytosis. 4. 5. Hydrolysis. 6. Which types of cell are stimulated to divide by the cytokines by T helper cells? B lymphocytes and T killer cells. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 7. Which statements correctly describe lymphocytes? 1. Some B lymphocytes and T lymphocytes become memory cells. 2. Plasma cells secrete antibodies to the blood plasma. 3. Some T lymphocytes stimulate macrophages to kill infected cells. 8. What is correct about the role of memory cells in long-term immunity? 1. They divide to form plasma cells and memory cells when the pathogen enters the body a second time. 2. They produce a fast response so that the person infected with the pathogen does not become ill again. 3. They remain in the blood and lymphatic system after the pathogen has been destroyed. 9. What are the functions of T lymphocytes? Production of cytokines, production of toxins and recognition of an antigen bound to an antigen-presenting cell. 10. A blood cell count can indicate how many white blood cells there are in the blood. Where else in the body are white blood cells found? Bone marrow, lymph and lungs. 11. Which row correctly describes B lymphocytes? They are not processed in the thymus and do not release antibodies immediately after their formation. 12. Which are specific immune responses? Production of antibodies add production of memory cells. 13. Where are antigens found? On the surface of pathogens, on the surface of macrophages but not present in the blood plasma. 14. Some children are born with severe combined immunodeficiency, SCID. Children with this inherited disease do not normally have any T lymphocytes and suffer from many infectious diseases. How may these children be cured from SCID? Treatment with stem cells. 15. Which sequence of events occurs during an immune response? 1. Recognitions of non-cell antigens. 2. Mitosis of B lymphocytes. 3. Development of plasma cells. 4. Secretion of antibodies. 16. Which statements about endocytosis are correct? 1. It is a passive process. 2. Materials are taken into the cell. 3. Vesicles form within the cytoplasm. 17. What is the first response by the immune system to a pathogen? Production of antigens. 18. B lymphocytes and T lymphocytes are often unable to respond to the antigens on pathogens that are intracellular parasites. What is the reason for this? The antigens are constantly mutating. 19. Which of the statements could describe both B lymphocytes and T lymphocytes? 1. They contain specific protein receptors in their cell surface membranes. 2. They divide by mitosis. 20. Which statements about macrophages are correct? 1. More are found in tissues such as the lungs than in the blood. 2. They have a role in antigen presentation. 3. They can engulf pathogens, dust particles and damaged body cells. 21. Which event will occur following antigen-antibody binding? Agglutination of bacteria to reduce their spread. 22. Which of the responses are correct for B lymphocytes made by cells of the immune system to a pathogen? 1. Mitosis. 2. Bind to infected cell. 3. Produce memory cell. 4. Secrete antibody. 23. What describes the function of a T lymphocyte? They can leave the blood and accumulate at sites of inflammation. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 24. What describes a nonspecific immune response? Ingestion of a bacterial cell by a neutrophil. 25. What are the functions of T lymphocytes during an immune response? 1. Destroy infected body cells. 2. Differentiate into memory cells. 26. Which row correctly identifies the roles of B lymphocytes and T lymphocytes? Secrete antibodies. B lymphocytes secrete cytokines. T T lymphocytes, provide humoral response, B lymphocytes 27. Which statement is correct for a neutrophil? They can leave the blood and accumulate at sites of inflammation. 28. What describes a function of a macrophage? It is found in tissues and secretes cytokines in response to infection. 29. Which action is taken by a B lymphocyte activated by an antigen? It divides repeatedly to form a clone of genetically identical plasma cell. 30. Which cells become memory cells in the immune response? B lymphocyte and T lymphocyte 31. Which is specific immune response? Production of antibodies. 32. What are antigens? Non-self macromolecules that trigger an immune response. 33. During a primary immune response, which order will be followed by the events? 1. T helper cells identify a specific antigen. 2. T helper cells secrete cytokines. 3. B lymphocytes with the specific cell surface receptors divide repeatedly by mitosis. 4. Some B lymphocytes form plasma cells. 5. Specific antibody is produced. 34. Which is correct sequence of events when a phagocyte responds to the presence of a pathogen, first to last? 1. Endocytosis 2. Vacuole formation 3. Enzymic digestion 4. Exocytosis 35. What are the functions of plasma cells during an immune response? Secrete antibodies. 36. Which statement about both B lymphocytes and T lymphocytes is correct? They produce memory cells to respond to an antigen when exposed in the future. 37. A graft tissue, such as skin, from a different person is usually rejected by the body. Which statement about graft rejection is correct? The graft is rejected by T lymphocytes because they circulate in the blood and can gather at the graft site. 38. Which row correctly matches a function with B lymphocytes and T lymphocytes? B lymphocytes provides a humoral response, T lymphocytes do not provide a humoral response. Structure of antibodies. 39. Where are antibodies found during an immune response? They are found on surface of pathogen and in blood plasma. 40. Rabies is usually a fatal disease. Rabies is transmitted to the people in the saliva of infected animals. A person who has been bitten by an infected animal needs immediate treatment. The treatment is a vaccination against rabies and an injection containing antibodies to the rabies antigen. What type of immunity is given by the injection of antibodies? Artificial passive 41. Which statements could be used to describe enzyme molecules and antibody molecules? Hydrogen bonds stabilize the structure of the protein and are important for it to function efficiently. Lecture notes for AS Level Biology by Muhammad Rashid Chattha. Cell number 03004610-457. 42. The tertiary structure of the protein molecule plays an important role in the functioning of the protein. How many variable regions are there on typical Y-shaped antibody molecule? 443. Which statement about the properties of the antigen binding sites in antibody molecules is correct? They have variable amino acid sequences for different antigens. 44. An enzyme hydrolysis the two heavy polypeptide chains of an antibody molecule. The hydrolysis occurs at the hinge region and breaks the antibody into 3 fragments. How many of these fragments are able to bind to antigens? 245. A student wrote down 3 statements about antibodies. 1. Their structure depends on peptides, hydrogen and disulfide bonds. 2. They are protein molecules with both tertiary and quaternary structure. 3. 4 polypeptides provide 4 antigen binding sites. Which statements are true? 1 and 2 only. Monoclonal antibody. 46. In the hybridoma method, what is grown by cell culture to produce monoclonal antibodies? Clones 47. What explains why monoclonal antibodies can be used to target cancer cells? Cancer cells have different antigens from normal body cells. 48. What explains why monoclonal antibodies are useful in the diagnosis of disease? Their shape is complementary to one specific antigen. 49. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. 3 types of cells are used to produce monoclonal antibodies. Which cells involved in this technique contain active telomerase enzyme? 1. Cancer cells undergoing uncontrolled cell division. 2. Hybridoma cells. 50. Which sequence summarizes the hybridoma method for the production of monoclonal antibodies? Inject mammal with antigens, harvest B lymphocytes from spleen, fuse with cancer cells, test for wanted antibody, large-scale culture. 51. Which row shows the cells that are able to divide continuously and are involved in monoclonal antibody production? Cancer cells, yes. Mouse B lymphocyte plasma cells, no. Hybridoma cells, yes. 52. The statements describe ways in which different types of monoclonal antibodies can work. 1. Binding to proteins on cell surfaces and triggering the immune system. 2. Blocking molecules on cell surfaces that inhibit T cells. 3. Blocking cell signaling receptors that trigger cell division. 53. Hybridomas are used as a basis for the production of large numbers of monoclonal antibodies. Fusing B lymphocytes with cancer cells. 54. The hybridoma method is used for the production of monoclonal antibodies. Which 2 types of cell are used in this method? Tumor cell and B lymphocyte. Types of immunity. 55. Which type of immunity does a baby have at birth? Passive, natural. 56. Which events result in a person developing actively acquired immunity? Becoming infected by TB bacteria and receiving an injection of antigens. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 57. Which row describes passive immunity? Do not trigger by an antigen, do not involve an immune system, memory cells are not produced and do not have permanent protection. 58. Which method of gaining immunity can be described as natural active immunity? Inhaling the chickenpox virus. 59. Which row commonly describes the type of immunity gained from being injected with an antitoxin? Not active, not natural but artificial and passive immunity. 60. What describes artificial active immunity? Stimulation of lymphocytes by antigens contained in a vaccine. 61. Newborn babies have natural passive immunity. What is correct for this type of immunity? This is temporary immunity in which antibodies are broken down. 62. A vaccine is used to create artificial active immunity. After being given a vaccine, it will take a period of time before a person develops long-term immunity against the disease. Which statement about this period of time explains this delay? No memory cells have been produced from B lymphocytes. 63. Smallpox was eradicated from the human population by a worldwide preventive program. Which type of immunity was triggered in people who were treated as part of this program? Artificial active immunity. 64. What occurs when active immunity is artificially induced? Self-antibodies attack non-self-antigens. 65. Which statement about immunity is correct? Artificial active immunity lasts for greater length of time than natural passive immunity. 66. Which statement describes how passive natural immunity is obtained? Antibodies are passed from mother to developing baby. 67. What describes natural active immunity? Stimulation of lymphocytes by antigens on the surface of invading pathogens. 68. What describes artificial passive immunity? Protection against a pathogen by an injection of antibodies. 69. The synthesis of specific antibodies in response to vaccination is an example of which? Type of immunity. Artificial active immunity. 70. During an outbreak of a very infectious disease, vulnerable people need immediate protection. Which type of immunity will be given to these people? Artificial passive immunity. 71. Which type of immunity occurs following infection by a pathogen? Natural active immunity. 72. An antiserum to a snake toxin can be obtained by injecting the toxin into a horse. The antiserum is made from blood plasma taken from the horse a few weeks later. The antiserum is injected into a person who has been bitten by the same species of snake. Which type of immunity occurs as a result of using this antiserum? Artificial passive. 73. Which type of immunity occurs as a result of the ingestion of antibodies by an infant through its mother's milk? Natural passive. 74. Which statements concerning the defense of the body against infectious disease are correct? 1. A specific immune response involves activation of B lymphocytes and T lymphocytes following recognition of, and binding to, a specific antigen. 2. Following invasion by microorganisms, natural active immunity can be gained by initiating an immune response. 3. Lysosomes fuse with vacuoles that have been formed by phagocytes and which contain invading microorganisms. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 75. Which is correct description of different types of immunity? Natural active, memory cells form; natural passive, antibodies cross the placenta; artificial active, injection of antigens; artificial passive, injection of antibodies. 76. Newborn babies have natural passive immunity. What is the correct explanation for this? Immunity is not inherited and antibodies are broken down. 77. To prevent a disease, dead bacteria may be injected into the body. What type of immune response is produced? It is not natural but active immunity. 78. Which statement explains why one-dose vaccination program for measles have not eliminated the disease? Some children need several booster doses to develop full immunity. 79. Why have vaccination programs eradicated smallpox but not TB? The smallpox virus showed little variation of antigens. 80. Which statements correctly explain why smallpox has been eradicated, but not malaria or cholera? 1. Cholera vaccines provide only short-term immunity. 2. Plasmodium antigens change during the life cycle. 3. Smallpox antigen remains stable. 81. The vaccination program for smallpox was a medical success story which resulted in the World Health Organization finally declaring world free from smallpox in 1980. Which statement correctly identifies the reasons for the success of the smallpox vaccination program? The virus was stable and not prone to mutations. 82. Whooping cough is a highly infectious disease of the gas exchange system caused by the bacterium Bordetella pertussis. Which method provides protection to infants against whooping coughs and reduces the chance of developing this disease later in life? Injections of dead Bordetella pertussis bacteria. 83. Why has it proved difficult to develop an effective vaccine against malaria? The parasites can only be attacked when outside the liver cells and red blood cells. 84. An influenza vaccine can be made by growing the viruses in chicken eggs. The viruses are extracted in liquid from the eggs and incubated. The purified egg extract containing the viruses is then used as the vaccine. What is a side effect of using this vaccine in some people? An immune response to egg antigens could occur. 85. The statements refer to vaccination programs for each of the diseases cholera, measles, smallpox and tuberculosis, TB. Which statement is correct for TB? The vaccine is not effective against some strains of the pathogen. 86. which of these statements explain why some vaccines can be taken by mouth but tuberculosis, TB, vaccine has to be injected? The TB antigens necessary to produce an immune response are proteins which would be digested in the stomach and small intestine. 87. Which type of new vaccine production would be most important in the fight to eradicate measles in developing countries? A single vaccine, without the need for boosters. 88. When a person is given a vaccination, immunity to certain pathogens develops. Which of the effects of vaccination are correct? Newborn babies have natural passive immunity. What is the correct explanation for this? 1. Results in artificial active immunity. 2. Stimulation of appropriate lymphocytes. 89. What happens when people are injected with dead bacteria? T lymphocytes produced antibodies. 90. Which statements explain why cholera has not been eradicated by vaccination? 1. People are mobile due to global trade and tourism. 2. The cholera pathogens target cells in the small intestine. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 91. Which explains why sickle cell is too difficult as they show no symptoms. Sickle cell anemia is an inherited disease, so vaccination is not possible. 92. The statements refer to polio and tuberculosis, TB. Which explains why polio vaccine can be taken by mouth but TB vaccine has to be injected. The TB antigens necessary to produce an immune response are proteins which would be digested in the stomach and small intestine. 93. Why has vaccination failed to eradicate cholera? The pathogen is present in the lumen of the gut. 94. Some children are born with severe combined immunodeficiency, SCID. These children do not normally have any T lymphocytes and suffer from many diseases. How may these children be cured? Bone marrow transplantation. 95. What happens when people are injected with dead bacteria? B lymphocytes produce antibodies. 96. Why is passive effective for only a short time? Antibodies are rapidly broken down. 97. What is first response by the immune system to a pathogen? Ingestion of the pathogens by phagocytes. 98. Where are antibodies and antigens found? On the surface of pathogens, antigens; on the surface of phagocytes, antigens; in blood plasma, antibodies. 99. The disease smallpox has been eradicated by a worldwide vaccination program. Which set of reasons correctly identify the problems associated with planning vaccination programs to eradicate other diseases? TB, different vaccine needed for active and dormant to active forms; malaria, different stages with different antigens; invades body cell; sickle cell anemia, genetically inherited recessive condition; cholera, invade gut cells where immune system less effective. 100. What is the difference between T lymphocytes and B lymphocytes in the immune system? T lymphocytes do not form plasma cell; B lymphocytes form plasma cells which secrete antibodies into the bloodstream. 101. Scientists are concerned that avian, bird, flu caused by the H5N1 virus could infect humans and cause a pandemic. If this occurs, which factors could help prevent humans spreading the disease? Reducing all air flights. 102. What are produced by B lymphocytes? Killer lymphocytes. Clones? No. Memory cells? Yes. Plasma cell clones? Yes. 103. After an immune response, memory cells remain in the blood for a long time. What is the function of memory cells? They divide to make plasma cells. 104. Which examples show the different types of immunity? Active artificial, immunity to smallpox after vaccination; passive natural, receiving antibodies to measles in colostrum. 105. Which future development in vaccine production is most important in the fight to eradicate measles in developing countries? A single vaccine, without the need for boosters. 106. What causes the measles vaccine to be less effective in children from less economically developed countries? Their diet does not contain enough proteins. 107. Newborn babies have natural passive immunity. Why is this only temporary? No memory cells are produced in the baby. 108. Why has it proved difficult to develop an effective vaccine against malaria? Mosquitoes have many stages in their life cycles. Lecture notes for AS Level Biology by Muhammad Rashid Chatta. Cell number 030046100457. 109. What is the immune system's first line of defense against invading microorganisms? Ingestion of the microorganisms by phagocytes. 110. For a period after its birth, a human baby is immune to most of the diseases to which its mother is immune. Which form of immunity does this represent? Natural passive. 111. The first breast milk produced by the mother for a newborn baby contains antibodies. What do these antibodies provide? Natural passive immunity.